Our guest speaker today is Shukla. He actually was an undergrad researcher with me at Cal Poly Pomona. So before I had my master's, before I had my PhD, while I was in the lab working on a master's degree, Viral was my undergrad research assistant, and he did great things there. Since then, he graduated with his bachelor's degree, and he is now at Cornell working on his PhD. He's just about ready for his first examination to move into a candidate, and then after that, a year or so later, he will be a doctor with work in academia. So he has a very colorful resume. He's done a lot of things in his time. A lot of volunteer work. He is with IFT, which I'm sure he'll talk about later. That's for food science specifically. So without further ado, I'm going to turn it over to Viral. Let's give him a nice warm welcome.
Thank you all for having me. Hopefully you can hear me all right. So today my talk will be innovation in a time of crisis using food technology and valorization to feed tomorrow. So a little bit about myself. I work at Cornell Agritech. It's just at the top of Seneca Lake. And as a food scientist and technologist, I work with chemistry, biology, physics, engineering, all the different scientific, mathematical disciplines, all related to food. So this could be food production, this could be post-harvest technology, this could be marketing, this could be sensory and human appeal to foods. So I'm just real close to you all, so please feel free to come by. I work both at Cornell Agritech and as well as Cornell down at the bottom of Cayuga Lake. So to start my talk, innovation in a time of crisis. Are we in a time of crisis? What is crisis? We've gone through a pandemic, we've gone through wars, global warming. Are these all pandemics? Are these all crises that we have to work through? How do these make you feel about the future? Are we worried? Are we frightened? Are we hopeful? What does the future hold for us when we have things like this? Nearly 75% of people our age, 16 to 25, are frightened about the future. They're worried about what's going to happen next. Think back to the pandemic when we were worried, is our food safe? Are we going to get COVID from just eating? There was loss of supply chains. We were worried that there would be no food. We all remember toilet paper, that was gone. War. In the war in Ukraine grows most of our wheat. With the war, that was disrupted. Wheat prices have gone up. Inflation, we all feel it. Every time we eat, every time we go out, we buy groceries, higher inflation. What does the future hold? And then finally, global warming. The climate is changing. That affects our food. It affects how we grow it. It affects some of my favorite things, coffee and gin. With global warming, those might disappear. So why, as a food scientist, am I talking about this? Global warming, crisis, what does that matter? Well, we all eat, and we all play a piece of this puzzle. So I'm going to tell you a little bit about my piece in this puzzle. So food creates a lot of greenhouse gases. About 25% of the global emissions is just for food, and you can see this breakdown here. A lot of it comes from supply chains, livestock, and then actual just growth of our food. Now, this is bound to happen. We have to feed people. We can't just go hungry. But populations are growing by the billions. How do we feed tomorrow? But 30% of calories are just wasted. They're lost. We do not eat it. We grow it, we spend all our resources, we put out greenhouse gases, but we do not eat the food. Further on, according to the United Nations, 13% is food loss and 17% is food waste. What does that mean? So let's take an apple. If you take the apple and you pick it, and then from there all the way to where you get your groceries, say Walmart, if you lose any part of that apple part of that way, that is food loss. This is mostly on the producers, the people who process the foods, people who create it. So you're asking, how do we lose part of an apple? Well, maybe this one was a little fungusy or a little grainy. We can't use it. It's a quality issue. We toss it. Maybe it's a safety issue. We can't use it. It's lost. Maybe you're enjoying a glass of apple juice. We took that apple and we squeezed the juice out. So the juice is the product, but the rest of the pomace is lost. Then there's food waste. That is once it is bought to the end consumer and then it is not eaten. Your apple's gone moldy. You don't like it. Maybe it's a little mealy. You're not happy with it, you toss it out. And that's fine. And it's usually a quality issue. It's hardly ever a safety issue because food put into intercommerce is safe. So my question is, how can we use food technology and valorization to decrease these numbers so we have enough food to feed the future while not creating more and more greenhouse gases? So I'm going to talk about 4 different projects myself and my colleagues have worked on that relate to this and see how our pieces of the puzzle fit in. So yogurt. We all eat yogurt, well, most of us. More specifically, spoonable yogurts. So Greek yogurt, you probably have bought Greek yogurt at least once in the last 3 months, maybe a little cup. According to Mintel, that's a data analytics company, 57% of consumers purchase it at least once in a 3-month period. When you make Greek yogurt, it's called a strained yogurt. That's because you remove the acid whey. You want that thick, high-protein yogurt dish, but then you're removing part of the acid whey. Can we use it for something else? Well, it's high in electrolytes. It has whey protein. It has a lot of great benefits, but it's acidic. It has a lot of fermentable sugars. It's hard to toss out. If we throw it into our oceans, it causes algae bloom. So me and my team have worked with acid whey and created various food products utilizing acid whey. Sports recovery beverage using it, ranch and cheese sauces, removing high quality, high economic values such as milk, removing those and using a low economic byproduct by valorizing it, giving a low economic product a value add. We've added to scones and packing mix instead of buttermilk, and we've also re-added it to yogurt to create yogurt beverages. Today I'll be talking a little bit more about the sports recovery beverage in detail. So this project, development of a sports recovery beverage utilizing upcycled yogurt acid whey by Shukla et al. When we run, we sweat. We lose a lot of electrolytes. And most of you will often reach for water or Gatorade, something that has an electrolytes in it. But what if we used acid whey instead? Make it a cleaner label. Acid whey has all these various electrolytes and nutrients. So that's what we did. And this here is a flow chart of pretty much what a food product developer does. We took acid whey, a raw ingredient, and treated it with activated carbon. This is our novel processing step in order to try to remove some of those off yogurt flavors and make something that's more palatable. But then we also have to do physicochemical tests on the acid whey itself. We take bricks, which is sugar, pH, acidity, color, all things that are necessary both from a chemical standpoint but from a consumer standpoint. We also do chemical engineering. We look at the carbon itself. We look at how it sequesters these different flavors. Does it attack other electrolytes? And then we do hedonic sensory studies. We did 2 in this case. We did one where consumers try the beverage and they tell us what they think. This is very important because if they don't like what it tastes like, they're not going to buy it. And that's the first rule of product development. But then since this is a athletic sports beverage, we had a set of students who would run on the treadmill for 30 minutes and then drink the beverage and see how it helps with recovery. And of course, we need to do economic analysis and willingness to pay studies. If it doesn't bring money, if it doesn't have a return on investment, there's no point in trying to valorize it. So some data. Here we see our pH and bricks. This is for the acid whey on your left and then the recovery beverage on your right. And we see non-significance across over 40 minutes of carbon contact time. This is good. This means that carbon is not taking away the electrolytes, the sugars, the things that we want. Here is percent salt and conductivity, also pretty much the same across. This is what we need. So how does it rank against other sports beverages? So it contains a decent amount of sodium, but a very large amount of potassium, which is very necessary in a sports beverage, but is often left out. If you look a little closer to our beverage versus Pedialyte and Gatorade, two recovery beverages that you might be familiar with, you see that when it comes to potassium, sodium, and zinc, we'll have similar amounts, slightly higher, slightly lower in some cases, but because it comes from dairy, we have phosphorus, calcium, and almost 25% of your daily protein just from a 12-ounce beverage. So this is a huge value add. That all of this is native to the acid whey. We're not adding protein, we're not adding nutrients. This is all native and would have been thrown away, but now we're creating more food. So did they like it? As you can see, we have the aroma liking. This is percentages of those who liked it and versus those who don't like it. In sensory, we use a 9-point scale because 5 is then in the direct middle. We have bottom 3, middle 3, top 3. So as you can see, SRB 0, sports recovery beverage with 0 carbon time, is liked far less than sports recovery beverage with 5 contact time and that with 10 contact time. But as you start to look at it, you see this BAAB. So what does that mean? That means that carbon is increasing the acceptability to consumers. We're removing that off yogurty flavor. But if you do it for too long, they tend to like it less. So that yogurty flavor, along with the little bit of lemon that we added, they like that. So that means that there is a more consumer appeal to using this. And overall, in preference, a lower number means that it's ranked 1 more often, or Sports Recovery 5 was ranked the best. When you test with our athletes, we tested against Gatorade. And as expected, Gatorade has a high suitability, but our sports recovery beverage was suitable for 50% of those who tried it, which is a decently high number, and it was more than double than what water was. And it actually had a 65% rehydration effectiveness on a 9-point scale. It's not bad. So with that, let's move on to grapes. Talk about evaluating grapes' effects of grape pomace on intestinal morphology. This is from Agrawal et al., one of my co-authors. So grapes, especially Concord grapes, are grown a lot in New York State. We use it for juice. If you ever had Welch's juice, it's Concord grapes. So when you juice a grape, you get a lot of juice, but you're left with pomace. Pomace is the leftover skins, the flesh, the stems, seeds, all that. It comprises most of the grape, actually, and it's usually tossed out. But what if we turned it into food products like acid whey? So this is the breakdown of how you make grape juice. You squeeze it, and then you get grape juice, which will be here GJ. You get GP, grape pomace, but you also looked at grape puree, GPR. Grape puree is kind of the mixture of everything. You can think of it as a control. So as you see here, TPC, total phenolic counts, antioxidants. Grape pomace has a very high amount of antioxidants. Most of it comes from the skin. You don't get that in juice. But then here, you see ADF and NDF. These are our fibers. And there's a lot of fiber. It's mostly fiber, actually, and then about 30% non-fiber carbohydrate. So this sounds like something really awesome to add. But what does it do to your gut? You know, it's a lot of fiber. We heard of prebiotics, probiotics. We need to examine this. So we used the Gallus-Gallus model. So the Gallus-Gallus, or your common chicken, the broiler chicken, we take the egg and we incubate it for 17 days. And then we take a water extract of pomace, juice, or puree and inject the amniotic fluid. Take the egg, inject straight into it, and then right before the chicken hatches, it eats the amniotic fluid, eats that yolk sac. And this is the first kind of meal, and in this case, the only meal it will ever intake. Now the key about the Gallus gallus is that it eats this amniotic fluid. So it goes through its entire digestive system. This is different than just injecting it or giving it to the egg. It is completely going through its stomach, the small intestine, large intestine, all that. And then we collect various morphological markers. So post-hatch, we see no significant difference. That's good. That means we are at a clean basis between no injection, water injection, grape juice, grape pomace, and grape puree. So what we see here is we took the microbiota of the small intestine, and we see that grape pomace doesn't disturb the gut microbiota. Here we see the first two, Bifidobacterium, Lactobacillus, and E. coli. These are in our gut. They may not sound like grape, but they live in there. And we see that grape pomace does not negatively affect our gut microbes. This is good. There is not a process where we're destroying them via dysbiosis. But we see in Clostridium, actually, we see that grape puree has a much higher Clostridium. That is not good. And that is mostly because of the fermentable sugars. So that means grape pomace has this kind of advantage, being that it's mostly fiber. Now let's look at various biomarkers. These are various genes and protein biomarkers for iron and zinc, and we see across the board no difference in uptake of these vitamins and minerals. And then we look here at the brush border membrane and vitamin A, and I want to point out VDAC right here, voltage dependent anion channel, and CRBP2, cellular retinol binding protein. And we see that they are upregulated. So this means grape pomace is actually increasing the ability of our intestinal lumen to uptake vitamin A and various other ions. This is most likely due to the polyphenols, also possibly due to the various fibers. But can we use too much? This is a plate of the intestinal lining, as you can see, of the chicken. And here we see villi and crypt cells. These are different cells in our intestinal brush border membrane in the small intestine. And we see a much lower number here. This means that the cells in the chicken were smaller, the depth was less, and they had less surface area. This is detrimental because we want more cells, we want larger surface area, because that's what absorbs all these minerals. So at 6%, there might be too much. And why is this? Well, probably because of the polyphenols, the antioxidants. Polyphenols can bind proteins and chelate them and stop them from being absorbed. So there's a such thing as having too many antioxidants. Let's move over to food technology. We talked about validation. Let's talk about high pressure processing. This is a high pressure processing unit we have at Cornell Agritech in the HPP Validation Center. This is what it looks like. Food goes into here and enters the chamber, and the chamber is filled with water. And it exerts high pressure on the scale of 600 megapascals. So it's kind of a large number. What does that mean? If you take your thumb, approximate about a square inch, and you pressed on it, you'd feel force. Now imagine a 140-pound person standing on your thumb. It's a lot of pressure. Now imagine 200 140-pound people standing on your thumb. That's the amount of force that this food is feeling right now. And it can be used for antimicrobial, it can affect enzymes, and it can affect all sorts of intermolecular processes. So let's look at two things, cucumbers and tofu. So first, increasing shelf life of refrigerated pickles through high pressure processing by Haney et al. Pickles, they're great, I love them. Crunchy, great with a sandwich. When you have a pickle, you want it to be firm, crunchy, especially without fungus. If it doesn't look good to you, you're going to toss it. Food waste. So can we decrease food waste by increasing the shelf life of pickles using high pressure processing? So we took two different kinds of pickles, two different brine pHs, left them untreated, HPP'd them, and then thermally process them. Thermally processing is what we typically use, shower pasteurization. That's the industry norm. And what we see here, at 400 megapascals and greater, we get no visible spoilage. So that is equivalent to thermal, if not better, but this is over 6 months. So first off, we have our first thing. The food is safe, it is pathogen-free, and it will not grow mold in your kitchen. Let's look at color. Fresh cucumbers are bright green. Well, on this screen, maybe not so, but bright green. But then when you heat treat them, they start to lose their color. They get pheophytin. It's more of an olive drab color. And what we see is from HPP, over a 6-month shelf life, the pickles are greener. They look healthier. They might taste a little bit better because you perceive greener as being more fresh. It also increases hardness. It affects pectin methyl esterase. It affects the enzymes in the pickles and causes them to get much harder post-processing. So you see here at thermal, it is quite soft. It is much less than an untreated cucumber. And then 200, 400, and 600 megapascals of pressure are equivalent, if not greater, crispiness, hardness. And this persists over time. So again, we've increased shelf life. Your pickles are going to be firm for a lot longer. So overall, if you high pressure process your pickles, they'll be greener, crunchier, and stable over time, thus decreasing food waste by allowing us to eat them for a longer time. This also gives us the ability to look at new types of products. The intermediate pressure gives the same amount of hardness, but a higher pH. So we can make less acidic pickles, which may be more enjoyed by the consumer. And then for our last one, enhancing legume protein functionality through high pressure processing. This is by Huang et al. Pulses, legumes. You've heard of them probably as peas, beans. They are very easy to grow. They're drought tolerant, they're nitrogen fixing, they're good for the soil, high in protein versus animal proteins or other proteins. Harder to grow, require a lot more water, create a lot more greenhouse gases. But people will probably prefer a steak over a can of beans. So what can we do to create plant-based proteins? So HPP affects non-covalent interactions. We have the native protein here, and then it goes through denaturization and then aggregation through the HPP process. This pressure physically destroys these bonds. And then afterwards, we see gelation with calcium. This is similar to, say, how you make tofu, but using only high pressure. Here it is broken down by pressure. We have our native protein. The quaternary structure is disrupted. Then water enters and swells, and we disrupt the tertiary structure. So this is a breakdown of the protein at the tertiary structure, and we can realign them. So we took protein concentrate, added to water, and then created these gels. Now, gel might not seem so appetizing, but pretty much most of the foods we eat are gels. They are proteins agglomerated together. And here we see that though they undergo a little bit more syneresis, they create this firm, chewy-like texture. Akin to tofu, but also almost akin to a meat that we're getting there. The key is, though, these proteins, they're affected differently from heat and HPP. Usually we can create these by heat, but heat affects different bonds than what high pressure processing creates. It's also heats from the outside in, while high pressure processing affects them all directly at once and affects only non-covalent bonds. So we see a much smoother texture compared to the very gritty, grainy texture we get from heat treated. So this means that we can use it to create very different products, whether it's a kind of a thinner gel, like a plant-based yogurt, or something a little bit more jello-like, like tofu, or even much firmer, because they cinnaries, a much firmer cheese. And with that, that is my piece of the puzzle. And I wonder what your piece of the puzzle is going to be, because nearly 75% of us are frightened about the future. I started off by saying that's worrisome, but maybe it's not so worrisome, because it means that we know what's happening, and we can do something about it. You learned what we have our plan for our future, so I wonder what you plan for your future to make it a little less frightening. I'd like to thank my collaborators, of course the various professorship, staff, and the various Cornell facilities, New York State Ag and Markets, and the USDA NIFA for funding. And with that, I'll take questions. Thank you. Yes.
Are you proceeding with any commercialization of, especially your sport replacement beverage?
Yes. So commercialization is a very important final step. At Agritech, we also work very closely with producers as part of our extensiveship. In about January, we'll be running all of our willingness to pay and economic studies. So based off of that, we will then look into proceeding if it would be a viable commercial beverage.
You mentioned like rehydration from that beverage. How do you measure that? Is that like something that the people tell you is happening, or?
Yeah. So we weren't able to run a clinical study. So a lot of this is from the consumers, and this is perceived rehydration. But there's also some factors because we had them run multiple times. We collected heartbeat and various other things like that. So through some calculation, we could get rehydratability. But a clinical study would be excellent if any of you are interested in running one of those.
So you mentioned tannins in the grape fungus, and you said that those are great for antioxidant, but tannins also produce astringency. I'm wondering how you do that particular mouthfeel in products that are researched.
Yes. Astringency, especially in grape skin. And then a lot of it comes from the grape seed. So one of the perks actually of the astringency is that it reduces the mouthfeel of acidity. So a lot of products have to be below 4.6 pH to be commercially safe, which makes it quite acidic tasting. So that tannin actually reduces it. It reverses that kind of flavor, and it helps with that. But when it comes to just pure tannic, the use of blending of various ingredients, we are food scientists, but in some way we're also cooks. We have to blend flavors and know how they work. And so proper blending, and then also the use of sometimes flavors. There's bitter maskings and various masking agents. Yes.
Did you do your summer study only with Concord grapes, or did you use any other varietals that are being grown in this area? Some of the wine, I'm thinking specifically some of the wine grapes, where again, tremendous amount of waste product potential that might have different properties depending on the
Whether it's red, black, or white, or just completely different varietals. This Gallus study was done on Concord because it was funded by the New York State Ag and Markets Concord Research. But we have also done actually a full study with chickens, and that is using various different kinds of wine grapes. So we're studying actually how fermentation that would happen on wine lees and must, how that also affects it. So it is a large growing project and a very much need, because yes, the wine industry produces lots of pomace.
I'll fire off another one for you. You mentioned you have 4 different projects, and they all sound really, really intriguing. One of the projects uses pressure in order to get rid of bacteria, yeast, I'm sure, all sorts of microbials. How does that affect the quality of the food, especially if it's protein you don't want to denature, or if it has some type of fiber component to it?
when we think about high pressure, we're worried sometimes of stuff imploding or exploding and all that. But the key with high pressure processing is because it's in a water environment, there is pressure on all sides equal and then also equal out. So there is very little destruction of cell matrices. Fiber is not very much affected. Proteins will denature, but just as heat, they denature, but then they will re-agglomerate in some form. So more segmented on that. Once you reach higher than, say, 10 to 15% proteins, they'll start to agglomerate, and if it's in a liquid beverage, they will fall out. We did a yogurt beverage, which we HPP'd as well, and there was no effect there. You can get a slight little bit more of syneresis, as you saw earlier, because it favors lower volume reactions, so that can cause a releasing of water.
So, when you're like measuring the proteins that are coming out, how do you what are you using to measure that? Like the proteins that are coming out of different things, or how they're being denatured.
So like the proteins after they've been HPP'd? Yeah, so we can look at it at the macro level and looking at how they create gels. So that's we have like texture analysis by cuttings and looking at how hard they are and all that. We can also use rheological techniques where we take a puck of the gel and you vibrate it through various frequencies and you get what's something called G prime and G double prime. And those are the elastic versus plastic. And this tells us how the molecules interact, so how they react, and we can tell a little bit more about the different bonds they've created, and then whether it's a gel or if it's not a gel based off of that.
Any other questions?
Taylor, I'll ask all the questions. Has crowd writ large, looking at making more shelf-stable products, looked at the acceptability to the consumer of the high-pressure treated versus, I'm thinking about irradiated foods to make them shelf-stable. Works great, but consumers resist because of misinformation or whatever. Have you looked at that? Are there any pushback emotionally about high-pressure processing?
Yeah, definitely. So we all know radiation, it's going to be scary, and a lot of it comes from the laws we put in. So radiation has to have that Radura symbol on it. HPP doesn't necessarily have to be labeled as high pressure. So you'll often find juices, actually, if you go into your market and you find like fresh green juice, cold pressed, all that, and you press the bottle. If it's kind of like a squishier bottle, that means it's been high pressure processed. So it's being done in industry. Actually, all of our sensory samples are worked on in Lidestri up in Rochester. They are a commercial HPP. Various products such as Ithaca hummus are HPP'd. You may not know it because it doesn't have to be labeled, and until there is a consensus that HPP is accepted by consumers, we'll just go on saying it's non-thermally processed.
All right, I have one final question for you. So, out of all of these, which one is your favorite project and why?
Project? Ooh. I like the acid whey. And the reason being, I was able to create like 8 different products with this acid whey, utilizing different components of what makes it good. Is it the electrolytes? Is it the acidity? Is it the yogurt flavor? What part of it? And then being able to learn different processing methods for each one. And I think that's kind of special with food science, is that we're given an ingredient and we can really take off and create very different products with very different processes and different consumer markets.